Good afternoon, everyone. Thanks a lot to the organizer for having me here and for just organizing this great conference. In this talk, we're going to be talking about micro-hydrology in roots and how it could interact with phenotypes. So you may know that water transport across plant tissues can go through many different paths. It includes the cell walls, cell membranes, and also plasmodesmata that connect the cytosols of different cells. And there's also an incredible diversity of anatomies. Just if you look at within the same root system, for instance, different root types, different levels of maturation, and it makes this whole system incredibly complex. And that's why I really like a good story to try to capture the central ideas of that. And in this exercise, I'm just going to propose 3 different narratives. And you will see that the first one involves gatekeeper cells and aquaporine. And I really like this word gatekeeper because it already carries some kind of story with it. And you will see that in a minute. And what we try to do with the model here is to make use of this concept of nexus that Graham Hammer presented yesterday. So we're iterating between the model and experimental data. And in this case, we tried to reject a complex hypothesis with the model. And the second narrative we're going to talk about is hydro signals and root development. And in this case, the model is used to dissect a process. So there are some variables that are really hard to put our hands on. And with the model, we can try to guess what they are and what's going on. And then we can maybe have more information about the process. And the first story is about the possibility that there would be a multicellular water pump inside plant roots. And in this case, we have been exploring the use of the model at the very small scale. And then there are surprising results that emerge. And the point now is to try to validate them experimentally. Yeah, now if you're ready for these 3 stories, let's go. Okay, the first story is about aquaporins. So you may know that these are protein channels that go through the cell membranes, and they're really key players for the water transport across plant tissues. And there's been some work going on in the past by Christophe Morel and also by François Chaumont. And you can see here in this specific figure, on the left, you can see the distribution of these aquaporins in hydroponics conditions, and on the right, in aeroponics condition. And you can see that there's a difference in the pattern. So in aeroponics, you have a lot of aquaporins that are also located near the epidermis. And there's another difference that's not shown here. It is that at the exodermis, you do have apoplastic barriers. And that's not the case here on the left side for hydroponics. So no apoplastic barriers and no aquaporins close to the root surface. And when François was trying to make sense of this data, he thought, yeah, maybe there's some kind of causation or correlation, at least, for the presence of these different patterns. And he also thought, well, maybe actually there's also something to gain if we were to force the expression of aquaporins in the last few layers on the hydroponics side. So you could imagine that if you were adding actually more aquaporins close to the root surface in hydroponics, then you would increase the radial conductivity of your root, and so maybe you can have access to more water in conditions where it's limiting. So. And he wanted to test that with mutants, and so he had this mutant to overexpress aquaporins constitutively across the whole root. And what he saw was very surprising. So he saw that actually at the cell level, the permeability of the membranes had increased, but not at the root level. No increase of the root hydraulic conductivity. And we tried to make some sense out of that with the model that's called MECA. And in this model, you can see that we have the anatomy of the roots, and then we can simulate water transport through the cell walls, membranes, and plasmodesmata. And what you see here in red, for instance, is high velocities of water, and then in blue, very low velocities. The cell layer that you can see here where I point to is the endodermis. So at the endodermis, there's no water flow through the apoplast because simply you have an apoplastic barrier, except at concentration points here where you can see that there's high velocities that converge. Those are passage cells. And so these passage cells, they don't have suberin at their surface, and so they're really like focus points for water flow. And there, aquaporins are really central players also to transport the water across the endodermis. And it's not the only location. So if you look in the model results, there's also a few cell layers before the endodermis that do take up water through their membranes and then transfer it on the other side of the endodermis through plasmodesmata. And with this model, we tried to test a few scenarios. And one of the scenarios was to consider that maybe aquaporins are actually expressed uniformly throughout your root. And then when you do this overexpression of aquaporin, you suddenly increase the permeability of all membrane at once. And we saw that in this case, actually, we always had a hydraulic phenotype for the overexpressing plants. So if that was the case, if aquaporins were expressed uniformly, you would have a larger radial root hydraulic conductivity. And the only scenario in which we did not see that was when your aquaporins, they were actually preferentially expressed and functional close to the apoplastic barriers. And that's really the location where we expect that they play these roles. And that also means that expressing these aquaporins where they were not originally there would not change the radial conductivity because just water is not passing this way. It's just passing through the cell walls, and then only when it has to, it goes through membranes. So that's the story we try to take up from these observations, and also in the model. Second type of like study case. Here we had a different type of phenotype, and in this case what you can see are tomato roots, and so they're initially in soils, and then at some point they reach an air gap, and in this air gap what you can see is called zero branching. So there is no lateral roots that grow in the air gap. And then as soon as you are back into the soil, there's more lateral roots that will grow. And this is a collaboration with Malcolm Bennett and Poonam Mehra, his postdoc. And what's great when we interact with Malcolm Bennett on this kind of phenotypes is that he always has these new technologies. And so this time he just came up saying, hey Val, check this out. I have this NLS abacus too. This is a reporter of ABA concentration inside the roots. And with this reporter, you can actually see in live the concentration of ABA inside your elongation zone of the roots. And you can see on the left that in contact with soil water, for instance, you have relatively low concentration in ABA throughout the elongation zone. And here on the right, you have much larger concentrations in white and purple of ABA. And then that means that there's suddenly, like within 1 hour, the ABA is all over the elongation zone. And Malcolm also had a few mutants, and from these mutants he could tell that this ABA is actually produced inside the phloem. And about the transport, he also had mutants with closed plasmodesmata, and he saw that the phenotype was not the same when we're closing the plasmodesmata. So we started to have lateral roots growing when the plasmodesmata are closed, which suggests that there's also a role for the transport of hormones through these plasmodesmata in the symplasts. And now we're trying to make sense of all these different mutants. And something we came up with was that yeah, maybe actually when we have transport through plasmodesmata, it's really unfiltered, unlike through membranes. So you have the water that's just flowing with anything that's in the cell sap, and that includes these hormones. And so we thought, well, if you have actually large water flow rates through these plasmodesmata, they're really like a focus point also for water velocities, then it's really hard for hormones to go against the currents of water. And so eventually, this field of water velocities is also going to determine the field of transport of hormones. And then we try to make a connection between this kind of narrative and the direct environment of the roots in contact with water or not. And we came up with the idea that maybe when your environment changes from air gap to soil water, you also change the field of water velocity, and then you change the field of hormone transport. And we did simulations of that, and that you can see here. And on the top right, you have your root that is in contact with the water. And we consider that actually in the elongation zone, you need to feed your cells with water for them to elongate. So you need sources of water. And on top, you have water that's available close to the root surface. And you also have water that's available in the phloem, and that's feeding sugars and water. And both of these water contribute to the elongation of the cells. And you could expect that the cells that are at the periphery, they mostly rely on outer water, while the cells that are in the stele, they mostly rely on phloem water. Now, once you are in this air gap, there's no outer water anymore, and then you can expect that all the water that feeds cell elongation comes only from the phloem, and it comes with all the messages that are including in the phloem, including ABA. And so that's the story we came up with. We have simulation that suggests that, yeah, it could be happening this way within a few hours, which is also the response time of like ABA transports in this data that you see on the left. And of course, at the same time as we propose these narratives, we're doing our best to just discard them with new experiments. And we're also in this nexus of iterations between model improvements and experimental observations. So now our next point is to try to observe water flow rates, and there's technology to do that. So one technology that can be used is Raman microspectroscopy that really allows to monitor D2O movements at very small scale. And with this, we can maybe discard this new narrative and propose a new one, or we can show that maybe it's working really well. The third narrative that I wanted to show here is about the thermodynamics of water transport in plants. And for this scenario, actually, so I can tell you what's the current paradigm. The current paradigm says that when you have transport across roots from the soil on the left to the xylem on the right, it always has to be from high water potential to lower water potential. And when it's through cell walls, it's only related to the gradient of water pressure, so you're really pushing water from one side to the other. And once you go through membranes, then you rely also on the osmotic potential. So you're looking at the total water potential, but you always go downhill of these water potential gradients. Now, what has been observed by a few scientists is that under certain conditions, when there's very limited water available, plants are still able to take up water and seemingly against this gradient of water potential. And from the physics, it should only be possible if there is a active water pump through membranes, for instance. And this is the kind of thing that has been observed for nutrients, but not for water. For water, we only have passive water channels called aquaporins, but no water pump. So it really leaves us with a big enigma here that's been standing for the past 40 years approximately. And it really looks like, yeah, maybe the plants, they completely ignore the laws of physics. And of course, there should be a rational explanation for that. And we tried also to explain it with some modeling. And what was the most confusing is that when I tried to reproduce this experiment with my model, is that my model was actually predicting also this uphill gradient of water flow, even though I just put the laws of physics from the cell scale. So yeah, it really left me completely wondering what was going on with my own work. And at some point, I realized that maybe this is a good thing, because it means that we have all the pieces of the puzzle. We just need to put them together to try to understand what is going on. And we came up with one possible explanation that you can see here at the bottom right. In the model, actually, we had non-uniform osmotic potentials between the different cells, the living cells. And for instance, here on the left, you see that the cortex cell had a very high concentration in osmolyte, and therefore it's very turgid. And it is connected through plasmodesmata to another cell type that is here in the pericycle, and that is less turgid because it has less osmolyte. And due to the difference in turgidity, you can have pressure-driven transport of water from the cortex to the pericycle. And as you see that locally through plasmodesmata, it's going downhill of the pressures, so that makes sense also as compared to the physics. It's at the same time uphill of the gradient of total water potential. So that's really how you deflect this kind of limitation of the physics over here. Now, from the modeling, we need to go to the experiments, and what we are trying to do now in my team is to measure these components of the water potential in the soil, in the leaf, water or flux also. And then we are also really looking at these 2D maps of osmotic potential inside living cells. So this is a very complementary type of data as compared to what's been done in the past. And I'm very lucky to have a team that's helping me with that. And hopefully we can make this journey from this model to experiments. And of course, we will need help from many, many people. So maybe some of you will be there in the adventure at some point, and we'll be very excited about that. And also, at some point, there will be a way back to the model, and there's probably going to be also evolutions that we need to do with the model. And maybe some people from other disciplines will also show up. And I wanted to finish this talk with another map, which you can see here, and that's based on the work of Clément Saint-Cast. And this is about interdisciplinary work, and that we think is really, really important to be able to navigate through this network and nexus of models and experimentation. As you can see, people doing image analysis and phenotyping are really, really well connected based on this bibliographic analysis. And on the sides, there's crop modelers and functional structural plant modelers that are a bit less connected to phenotypers. And I was actually wondering, within this room, are there people who have been interacting like as phenotypers with people doing modeling in their own research, or maybe are there people doing modeling and who have been interacting with modelers? So maybe those people can raise their hand just so we have an idea if, like within this auditorium, there's people doing these kind of interactions. Okay, so I see a few of them. Maybe that's actually, it suggests that maybe, yeah, I think we're a bit biased, but of course, I think maybe it's also a sign that people like François and Wendy Silk did a great job bringing this kind of narrative of interactions between modeling and experiments. So with that, thank you very much for your attention, and I'm really looking forward to creating more of these narratives with you all.